In first two sessions, we have learned about some basics on bioinformatics, and then in particularly we have learned about genome informatics, where we learn about different algorithms that are used and different approaches and different problems that exist in domain of biology, which attracts computer science and people from mathematics to solve and develop methods that can help to solve those big problems. So in today's session, that is session 3, so we will talk about some big data that exist in biology, and recently these multi-omics big data have emerged, and they, as I will show you, that these data sets are really now huge in terms of storage capacity and also in terms of analyzing. So far, we have just looked at genomics data in last two sessions. But if you look in biology, we have multi-dimension data, multi-modalities. So for example, genomics is just one level. We can have data on proteomics, we can have data on metabolomics, we can have data on phenomics. So whatever that ends with this term, this word omics can be covered under this umbrella multi-omics. And so the challenge comes when you want to kind of integrate or draw the lines across these different layers of omics data sets. And in this session, I will show you what are the available methods and what are the ways what are different schemes you can use to integrate these different layers of data. And I will also talk about different strategies and what are the different ways this data is generated. All right, so I hope you were there in the first two sessions. So quickly, I want to show you this. So I'm Dr. Pankaj Yadav, and I am working as assistant professor at IIT Jodhpur. And I have obtained my PhD in statistical genetics from Germany, and my MSc is also from Germany. And I have a BTech from Punjab University in biotechnology. And currently, I am working in this multi-omics field, where we have data from genomics, transcriptomics, proteomics, and metabolomics. And we also work on integrating these different types of data sets. And with this, for this, we use machine learning approaches and statistical approaches that exist, and also we work on developing these methods to solve these integration problems. Right, so our ultimate goal is to do predictions, diagnosis, better diagnosis, and improve the phenotype predictions by integrating these different layers of data. But we know that there are many challenges in integrating these omics data sets, and I will talk about this in this session also. So the challenges start even from the experimental stages, where we have biologists working in lab to generate this kind of data, whether it is sample preparation, designing different study designs, and reproducibility, and calculating statistical power. These are all challenges that starts with the beginning of a study. And then we have individual omics data sets, and pre-processing itself is very big challenge, such as normalization, transformation, and imputation, because most of the time the data is unbalanced and a lot of missing values are available. So these are all preliminary challenges, and then the issues come when you want to integrate these different layers of data, like data scaling, reduction, and what are different ways we can integrate this kind of different levels of data. And then, of course, we have to develop methods to deal with the visualization, to make some biological inference is ultimately our goal, how we can help this kind of integration in biomarker discovery and then validation of targets and databases for usability and reproducibility, and then ultimately, as I was saying, the phenotype prediction. Okay, so I believe that most of you come from computer science background, but still I would like to take you through all these basic definitions so that you can follow up this session. So let me just start with this very basic definition. What is data? So data is basically a set of values of qualitative or quantitative variables about one or more persons or subjects. So it can have qualitative nature or it can be quantitative. For example, you can have the body heights of the individuals, or you can have, let's say, the body weights, right? And qualitatives can be your, let's say, conditions which are categorical, and you will find a lot of examples in biology itself. You can think even if has conditions like different conditions where you perform the experiment. And what is data set? Data set is basically collection of related set of information that is composed of separate elements that can be manipulated computationally as a unit. So it is collection of related data. So from data you are reaching to data set by collecting different related set of information. And now the next thing is database. So database is basically or data repository are basically the means to store the data. So once you have generated the data, where do you store? So there are many databases we already learned are also in the first two sessions, such as NCBI, Ensemble, and Uniprot, and so on. And so these are basically the virtual data storage that stores, organizes, validates, and makes accessible core data related to a particular experiment or system, or you can say experiment, or you can say set of experiments. Sorry, and so the metadata. Now, metadata is basically the data about the data. So data that describes your data. So for examples, you can have your experiment, and in biological experiments, you usually find the title of the experiment and abstract giving the overview of the experiment, and then authors and other keywords like publications, if any. Or you can also have information about sample collection place, origin of samples, or you can have origin sample submission dates and all such kind of information. So basically it describes your data. So we usually see workflows. So what is the purpose of what are workflows and what are their purposes? So we will also see in our coming session that there are few workflows that are available for analyzing multi-omics data. And basically these are defined as series of tasks or different series of, you can say, operations for processing your data. So starting from one point as an input to output. So you have a well-defined structure that does a lot of intermediary tasks on your data. Then nowadays a new, you can say, the hot topic nowadays is data science. So data science has exactly no definition, but there are many definitions that people have come up with. But in principle, it is an interdisciplinary field because it requires information on not only one domain, it requires information on many domains. And it is interdisciplinary in that sense. And also it is in this kind of field, we have this quantitative and analytical approaches, or we can have processes and systems that are developed and used to extract knowledge and insights from increasingly large and complex set of data sets. So this is just one version of the one, you can say, one definition that is I have given here, but there can be other definitions of this field. But the essence is that it is interdisciplinary. You need to have information about computer science. You need to have information about mathematics, mathematical methods, computational methods. You need to have a strong background also in domain-specific fields such as biology if you want to apply this data science in biology. All right. So deep learning is also probably most of us are aware of this, but still I want to just quickly tell you that it is type of network in which each successive layer uses output from the previous layer. It is basically inspired by biological neural networks, and it is similar to communication patterns in biological nervous system, where we have different layers and signals are passed from one layer to the another layer. And then we have omics. So omics, I think most of you are not familiar with. So it is a collective characterization. As I said, it can have data from different categories, or it can have different types of data. And omics is basically a collective term. It is collective term for characterization and measurement of pools of biological molecules that translates into the structure, function, and dynamics of an organism or organisms. Examples include genomics, proteomics, metabolomics, and other types of omics. Phenomics, for example, is also part of this omics. So I will talk about this in the next slides. So because this kind of this session is also aimed also to introduce you about biological data types, and also what kind of methods that are available to use that are used to analyze these different types of biological data. So genomics data we have already learned in our session 2, right? So where we have learned about nucleotide genome sequences, which are basically composed of 4 nucleotide bases, A, C, T, G. Then we can have metagenomics sequences also. These are different types of genomics data you can have in this category. And then what kind of work can be done, right? So with the genomic data, we have already learned that we can do sequence alignment, we can do comparison analysis, we can develop phylogenetic trees, right? So we can also do many other things. So only a few examples I have shown in our previous session 2. So apart from that, you can have one can do gene finding, means you can identify novel genes, so which regions of the this sequence is coding for some kind of protein or any other factors. And then you can also do a functional annotation, like similarly like you do gene finding, you can also annotate, or you can assign which regions of this genome is doing which kind of work, right, based on some computational prediction models. And then as I already shown, that you can perform sequence alignment, and then you can do homology determination, or you can do comparative analysis and phylogenetic inferences. Also, you can also perform association, right? So association means you can associate these polymorphism or regions where you find the variations in your genome with patients, if there are any patients that are having some specific kind of variations at particular positions in the genome sequence. So this kind of work can also be done. And then you can also do mutational function prediction and species distribution analysis based on doing some clustering analysis, following some clustering approaches. So this is something you can do with the this genomics data, right? And then the next layer of data that comes in biology is transcriptomics. So remember, we have learned about this process where the DNA is getting from DNA to protein, right? So how do we reach there? So first the DNA is transcribed into RNA, and then RNA is translated into proteins. So I hope you remember that. So next level of data that exists in biology is at transcriptomics level data. So where we have RNA expression levels. So basically these are the number of transcripts or number of you can say RNA molecules that are produced in a given condition, right? So, and a given condition or set of conditions. So it can be transcription factor binding data, or it can be chromatin structure information. So these are different types of data that you can have under this category. And what kind of analysis or what kind of studies can we perform in this kind of data, right? So one can perform a differential expression analysis. So compare two conditions, let's say in the day and night, which genes are expressed in daytime and which genes are expressed in the night time. So this is one type of study that can be performed. And then one can perform clustering to see if there are any groups of patients which have specific type of expression profiles. right? For example, you can compare patients with healthy individuals and see how these expression profiles or transcriptomics profiles are changing in are behaving or are occurring in these two kind of patients in these two groups of individuals, patients and healthy individuals, right? So one can also do functional enrichment and transcriptional regulation or causal reasoning, which gene is, or you can say, or which kind of transcriptional factors are regulating which kind of. So remember, it's biology a little bit complicated. So these RNA levels expression molecules can also go back and also regulate their own expression or expression of their other partner RNA molecules. So this is also possible. So you can also perform these kind of studies. And then you can have proteomics. So after this transcriptomics, you have next category of data, that is proteomics. So as I was saying, so from mRNA, you get to the protein level, and you can have different types of data that are available in proteomics itself. So you can have, let's say, proteins level. So again, you can measure which proteins are present in your samples. You can have proteome profile, or you can say you can also compare what kind of protein profiles is available for individual. So you can also look at the structures. So proteins have very unique structures. So depending on what kind of sequence they have, amino acid sequence, and from where they are in which part of the body or which part of the cell they depending on that, they can have different structures, primary, secondary, or they can have also 3D structures and also 4D by making complexes. And of course, protein interaction means exactly that you have more than one protein kind of involved in some function. So they may be physically interacting with each other. So let's say 2 or 3 proteins may be performing some function when they are only interacting each other. Individually, they do not perform any action or do not perform any function, let's say. So, so what kind of things can be done with this data? So you can do, first thing you can do is you can even do a novel protein identification. So if you see that, if you find any sequence and then you can just compare, or you can again do the alignment also with the protein sequence data to identify whether you found a novel protein that is been identified in your sample, or you can, and by comparing with the existing databases. So you can do that kind of study, or you can perform protein functional predictions. What kind of functions these proteins are doing, you can also study that. And you can also do what kind of structure given a sequence, you can do prediction analysis and find what kind of structures this protein will have. Or you can nowadays, this with the advancement of AI machine learning tool and deep learning tools, one can also predict the other way around, the sequences given you have a structure. So if you have given structure of the proteins, can you predict what kind of sequence it will have? So this kind of you can also compare 2 or more structures, how similar they are, or in which regions they are different. So structural comparisons based on graph theory has also been done. A lot of work has been done in this field. Then one can do molecular dynamic simulation. One can simulate the behavior of proteins in a given system. So one need to simulate the whole system artificially with the help of by including different there are several tools like GNOME now, they are available, where you can simulate the biological conditions and you can see how the protein is behaving, how is it folding or unfolding, and if you what happens to this structure, or what happens to their behavior when you change different parameters, right? So this kind of dynamic simulations can also be performed with this kind of data, and you can also perform mutational functional predictions. You can mutate 1 or 2 positions on amino acids, and you can see what kind of functions will it have based on these kind of mutation profiles. So given this all, you can also perform docking predictions. Basically, that means that you can see which part of the, or which drugs are fitting well to the protein, so that they can be these proteins can be inhibited. So what happens is in certain disease conditions, certain proteins are produced, like in case of like antibody reactions or immune responses. You can perform those, you can find those inhibitory molecules that blocks to the protein, and the function of the protein is inhibited. So protein is no more functional by doing or by binding into their pockets. So these drug molecules act by that, and one can do docking procedures or docking predictions to identify which drugs molecules will fit, or which molecules will kind of inhibit the function of the protein. Or one can also do this protein-protein interaction analysis to identify which set of or group of communities of proteins are acting together for the functioning of some biological function. Right. We will also be covering in this in second session. And then we can also have this metabolomics data, where we have basically the small molecules, or you can say the chemicals that are biomolecules that exist that characterizes, or you can say different pathways, let's say glycolysis, or this tricarboxylic acid, cyclic acid TCA cycle, where you perform how this our foods is broken down from glucose to how you generate energy. So these kind of chemical or biochemical reactions that are happening, the data on those reactions is available, and one can perform pathway and metabolic network analysis. Right. So you can study whole pathways such as glycolysis, and just let's say if you want to let's say you interpret how these molecule or pathways can be blocked, or how you can be inhibited to avoid, or to, you can say, the generation of different molecules. So there are many other studies that can perform with the help of this metabolomics data. And apart from this, nowadays the imaging data has become very popular, very popular. So most of the work that is going on deep learning is based on this machine learning on this imaging data in biology. So you can have microscopy images, you can have MRI images, you can have CT scan images, and other images also. So what can you do is you can do feature extraction, and you can also do high content screening based on this imaging data. You can also do diagnosis, or you can do prognosis analysis. And cytometry also, you can have data on cell levels or cell phenotypes. This is a little bit also new advancement, where you can cluster cell specific population. Cell means this biological molecule cells, single unit of life, where you can also identify different biomarkers to you can say to stop the growth, or you can say if you can identify some anti-cancer molecules that can be used for treatment of the patient. And then system biology. So this will again, it will be a new topic, and I will be discussing it more in the next coming session, but many things can be done also in this kind of field. So if you think in terms of the storage capacity or physical storage that is required for this kind of biological data are very, as you can see, the complexity is very huge. So this is nothing new for the computer scientists, but still, if you think of storing this biological data, you need to think from terabytes, beyond terabytes. Beyond terabytes to you can say exabyte. So this is somewhere you need to think about when you are thinking about storing this big multi-omics data. Now, so as I was saying, this multi-omics field, it's interdisciplinary, totally interdisciplinary. One individual cannot have entire understanding of this kind of field. You need to have a close interaction. You need to have a close collaboration between biologists, computer scientists, statisticians to really solve the true biological problems. Because so this slide actually tells you what are different data levels in biological research. So starting from let's say from mice. So this is just an example. So where you collect the, let's say, blood sample, or after this you transfer it to this Petri disease, where you have you are probably checking for some anti-cancer or any other kind of growth functions in this. How, and you can store you can do many things with this kind of sample in laboratory. And after this you can, for example, one possibility is that you would like to check the gene profile, or you can say the gene expression in a given condition. You can have different types of Petri dishes depending on which conditions you have. And after this you can pass on this to some machine, or you can say these Illumina scanners, where you can basically perform what kind of data is and what kind of expression profiles is produced. So basically you use this microarray chips that we have already talked about in second session. So once you have this, you will see that from this machine, basically it will scan these chips, and it will give you this kind of pictures. So this picture, I think we already also talked about briefly. So basically these different colors refers to whether these conditions, whether these genes are expressed or underexpressed or overexpressed compared to a reference condition. So you can compare, let's say, healthy individuals with the patients, and then you can see, okay, how this gene expression pattern is changing in these patients. So from here to here, let's say, you can say here mainly the biologists are involved, those who work in the wet lab, and then from now onwards you can say mainly the bioinformaticians, they start working here. So they can analyze, or they can sequence, or they can produce this kind of imaging data using with the help of these machines. So after this, you can say the work of bioinformatics is also there, but mainly now the computational skills are required. So we have now imaging data, and from this imaging data we need to have some algorithms which can convert this imaging data into this textual form, so text files, where it is readable more in the form of text. So this is called derived data. So a lot of algorithms already exist to get this kind of conversion or transformation from image to the text data. And now this is mainly now analytical job that is left to statisticians and also mathematicians, also computational biologists. So here now you have to make sense of this data, whether you can, there can be many things that can be done with this data. You can study gene expression profiles individually, or you can compare the let's say the profiles among different groups of individuals, or you can perform different clustering approaches, or many other things, such as you can also do this pathway analysis. You can do many other things that we have talked about. So yeah, so after this again you have to so you have to come back and you have to interact closely with the biologist to understand or to make some sense of these observations that you will have. So basically you will have is p-values, or you will have some sorts of clustering, and then you have to need to have domain experts who can explain or can make sense out of this analysis. So see, the idea I'm here is to to tell you that you need to have a interdisciplinary team of people to perform this kind of work. You need to have biologists, you need to have bioinformaticians, computer scientists, math statisticians, from this step to this step. All right, so this is something that I already shown you. So again, this data comes from NCBI, and you will see that how the you can see different types of data now that has grown, has really become exponential in terms of, so these figures are now in millions, you can say. So we have more than now 250 million sequences that are available, and a lot of other data that not about from sequencing data, but other kind of data is also available and easily accessible from NCBI, open to everyone. So yeah, so I think you already know this slide. And so I just want to show you this NIH strategic plan for data science, because data science in biology is also like gaining a lot of importance. A lot of people are now moving towards this field and trying to pursue their career in this field because the opportunities are really high. So they have come up with this policy, you can say, or strategic plan that they have proposed for doing any kind of data science work in this field. So they have proposed a policy for data infrastructure, which basically focus on optimizing the data storage and security, which is one of the important tasks. And then they also have provided this connect NIH data system to make this, and they have their own data infrastructure, and they have connect NIH data systems. And they have also created a new scheme or strategy for modernizing this data ecosystem, where basically they modernize this data repository ecosystem and then also support the storage and sharing of individual data sets. So sharing and storage are important terms. And then we have this, you can say, better integration of these different layers of data, how we can integrate clinical and observational data for biomedical data science. And then the management and analytics and tools that are available for analyzing this kind of different types of data. They have provided a policy to support useful, generalizable, and accessible tools and also workflows that are available from this NCBI portal. And then they have also broadened their utility of and access to specialized tools which are specific for doing some specific work actually. And then they have also policy to improve the discovery and cataloging of the resources, which is also an important thing. And then again, so workforce is another important requirement, because if you don't have that to work on this data and developing these kind of communities who can work on this direction or in this domain. So they have a policy to enhance the NIH data science workforce, and they are planning, or they have actually implemented, to expand this national research workforce and engage a broader community in doing this kind of data science in biology. And they have, of course, stewardship and sustainability policy, and they have developed this policy for a fair FAIR data ecosystem. And also they believe that enhancing this stewardship is one of their key policy or key plan. Now, to give you some examples of this multi-omics big data from where you can find. So one of this example that I have just shown in this slide is from Omics Discovery Index. So basically this is a data repository where you have all kinds of multi-omics data that is available, right? So you can have data from genomics, you can have data from transcriptomics, you can have data from proteomics, metabolomics, all these kind of different types of data that are available that can be accessed from this single platform. So this is very interesting and a very novel tool that I think started in around 2018 somewhere. So so you can see this this metabolomics data is rare to find, but most of the other kind of data like genomics and proteomics, you can find these are most abundant data. And then you can also search this data set by clicking on the different organs. So you can click on organs, lungs such as liver, or you can heart, whatever. So you can look for, or you can filter by organ specific data, or you can also click, or you can also look for similarly you can look for organism specific data, or you can look for disease specific data such as different cancers. And you can so these are, they also like very quite updated, they keep themselves very updated, and they also keep on highlighting on their homepage what kind of new data that has come to this repository, right? So for example, here you can see they are June 26th, the FLNC D1821 bio ID experiment was done, and this data is now been available, and so on. So this is very you can say very live, or you can say they are people are actively involved in enriching this repository. And mind you, so this community is also now really big. A lot of other people have now being involved in developing, or you can say making tools on this repository. So quite an interesting platform if you are interested in working in this field of multi-omics big data. So the next example that I want to show you about this big data in biology is this cancer genome atlas. Basically, it has you can say it has almost all kind of cancers that exist in humans. Their data is available from this portal, and you can say I just picked it from their website. So the next dozen years, TCGA generates over 2.5 petabytes of genomics, epigenomics, sorry, and transcriptomics and proteomics data. So you can say this the level, or the complexity, or the storage complexity required, or the amount of challenge that will be faced by coming time in handling this cancer data itself is a challenge. So this data, which has already led to improvement in our ability to diagnose, treat, and prevent cancer, will remain publicly available for anyone in the research community to use. So this is really a very nice source if you want to work in this cancer research and want to study, or want to apply computational methods, or want to develop computational methods to help in treatment or diagnosis of the cancer. So you must definitely have a look on this TCGA portal, because the data available is really curated, and you will find almost from across the world, you will find data deposited by different people, different scientists. And also not only this, they also have running projects. So if you are interested in proposing some projects, they are also funded. So you can apply for funding and get your project approved, and you can this project will be then highlighted, or you can say there will be also possibility to collaborate or generate collaborations. If you just go to their portal, you will find more details about that. All right, so next example that I want to show you in this field, big data in biology, is about this human cell atlas, which is actually one of the recent trend, we can say, or new theme that has emerged. So far, we have been treating the whole human body or whole organism and trying to understand the or system, or you can say the whole body as a system. But now people have changed their themes, and they have now become a huge community has now been involved in generating this single cell multi-omics data. So what does that really means is, so you treat each single cell as a system itself, and you try to understand how these different cells are behaving in your body, right? So it has been observed that a lot of studies have found that given within a like, so what you, how the organ, how you like categorize the systems is you can have human body, and within body you can have organ system, you can have tissues, and within organ, and then you go to the cells. So within as a given tissue, which is actually comprised of cells of same type, or you can say, so tissues within same similar within same tissue which has same types of cells, even they have different profiles of different expression profiles, so they also differ from each other in respects. So so now the idea is to like look at those cell and look at this cellular level and try to understand how this, what are the differences between cells of same, they belong to same tissue, but why they are behaving differently. So a lot of data has been generated, so you can see these are some recent numbers that are provided in on their portal, data portal. So they have data on about 4.5 million cells now, and they comes from 33 organs in humans, and so far only around 289 donors have been enrolled, and they have their data has been collected. But you see, if you look at the cellular level, even the data from 10 samples will be really huge, because you have millions of cells to analyze, right? There are currently again 28 projects are working on, and they also conduct. They are also very active in this kind of research, and they organize a lot of workshops and seminars and conferences to to attract people or to invite people who will be interested on working on those projects. And there are many other almost 81 labs that have been associated to this community across the world. So if you have interest in working in this single cell multi-omics and data science, so please have a look on this HCA portal. All right, so this I think I have already discussed in session 1. So I think I will skip this. So this basically tells you what is nucleotide sequencing and how this sequencing works, and how traditional Sanger sequencing has been replaced now by next generation sequencing technologies. So yeah, so this is then again some repetition from, or you can say the revision from the previous session, where it shows you the how the sequencing is done. So you basically start with this large DNA molecule, and then you fragment it into small pieces, targets, and then you sequence these small pieces, and then you perform the assembly, right? So this is something we have already talked about, and then you come up finally with the final sequence representing your target molecule. So there are a lot of algorithmic work that is involved here, and mainly in this from here onwards, from assembly to then continuous assembly. All right. So as I was saying, this next NGS is basically very recent, where you have this high throughput techniques to sequence this data. So you can have your whole sequence get sequenced even in less than $100 nowadays, and one can one can store this into their computer memory or system, and perform also the analysis. So this is time where we will, in coming years, we will see that each of us have our DNA sequence available, and we can perform analysis on our own data. So this is going to be the reality in future. And I was saying also, I think I already mentioned that nowadays this personal sequencer also emerged. So this can be used to even analyze or sequence the data outside the laboratories, right? Outside the laboratory means Usually the sequencing will happen inside these wet labs where we have experimental labs. But nowadays, very recently, and those sequencing sequencers are available. You can take them outside. You can take them anywhere you want. You can even do in sequencing. In the I think in the coming few years, you will have sequencers which can be used to sequence the live organisms under the water or in a given soil sample. So that kind of work is also going to be reality in few years. And you can imagine how much data then it will be generated. So far we have the technologies, so we have to, so what I want to say is that you need to have a pace with the kind of technologies that are we are expecting in coming years, because the techniques to generate the data has already like got really in the advanced stage, and in coming times it will also go to a higher level. And we need to understand, or we need to develop methods, we need to develop mechanisms to handle this huge amount of data and making sense of those data. So this is something we need to keep in pace with the these technologies that are emerging. And now, so again, coming to this multi-omics field, where we have been talking about how we make sense, right? How to make sense of this kind of data. So, as I was saying, that we need to have a close interaction between a statistician, bioinformaticians, computer scientists, and molecular biologists to provide meaningful results. Without that, understanding or solving any problem in this field won't be possible. So yeah, so we here again it's just a cartoon which shows that get all the information you can, and we will think of a use for it later. So we need to have this kind of understanding, right? So we just not to just compiling the data. We can have unlimited quantity of data from multiple and heterogeneous sources. This was also I was talking when I was saying that we can have our personal sequencer. So everyone even can have their own sequencer. They can keep it at their home, or they can use it for sequencing any kind of organism that it will happen in the coming time. But what is important to emphasize here is that we also need to have computational issues to foresee, right? Otherwise, this generating this data will not make sense. Just it will lie, and nobody will be caring about this. So storage is one thing, and then you need to have some computational ways to make sense, right? And after that, you also need to have a biological interpretation for validation, right? So we need to keep a pace with these new technologies. So far, we have in biology if you talk about before last 30 years or last 3 decades before that, most of the work has been focused or people have followed this reductionist approach, where they have looked at not even a single gene, but maybe part of a gene. So people have been focused. They have spent their life studying even a single gene. So this has happened in past. Now, and they probably exploring the single gene and try to understand what are the functions, what are the sequences, what can be the different mutations and things like that. So people were focused mainly on this single gene approaches, and where they have only one hypothesis to test, and they were mainly restricted to only few statistical tests to perform. So this is a thing of past, and now we need to adopt a holistic view. Means that we need to analyze now thousands of molecules, thousands and millions of molecules to make some sense. We need to have some hypothesis-free tests also to be performed, and not only just focused on one kind of data. We need to understand a complete understanding of the system. We need to have understanding of how the system is interacting with environment. What are different biomolecules? what are this genomics, what is transcriptomics, what are proteomics, and how these signaling networks, or we can say, you can say networks that biological networks that exist within the system. And also now, as I was talking about that, people are also now focused on analyzing single cells to study or perform kind of work and looking at single cell level. And then we can also have understanding of organisms, how different organisms are arranged or they behave. And of course, we need to, no doubt that we need to have close interaction with clinicians, because they are the one who will be able to and finally translate our research into practice. So this all need to be keep in mind before when we in today's scenario we need to have this holistic view. We need to involve all these factors in our studies. So as I was saying that so in most of the work that happens in this multi-omics big data is basically the integration or making sense from one layer of data to another layer. And also we need to have, of course, individual level understanding of different at the individual layers. So for example, we need to have understanding from transcriptomics to proteome, and then we need to have understanding of metabolome, and probably also very new topic, or not new, but last few years only it started, where people are, most of the work in European side is now in Europe and also in America is going on, studying the host microbe interactions. How does our microbe composition in our gut is making us healthy or unhealthy, and how does our microbiome inside and outside affects our environment. So these kind of things that are also done. And then we have on top of that the clinical data. So we need to like kind of draw a holistic approach to integrate not only this meta-omics, multi-omics data, but also we need to consider all other aspects of this research. All right. So as I was saying, so this figure here just show the same thing that I was talking a few slides back, that we need to have an interaction between, we need to kind of integrate these different types of data. So let's say we have genome, and then we have transcriptome, and then metabolome, and then proteome. We need to understand where are the connections. So how we can connect one layer, where we make connections from one layer to another layer, right? And these are shown by these dotted lines, where you have this probably this gene probably is making or maybe associated with this protein, and sorry, this mRNA, and then probably this gene might be associated with this protein. And probably if we change or we delete this, you will probably also see that this protein is also affected, or it is existing no anymore, or its function has changed. So we have to perform these kind of studies, or we need to draw these lines across these different layers. And so we need to develop these omics relationships, but these are not that straightforward. We need to have understanding of many things in order to draw these lines across the layers. So I have shown you some examples about multi-omics data, and this slide show you some more examples from where you can get additional data, multi-omics data, particularly on cancer. So you can, because cancer research is one of this biggest topic in biology, and many institutes and laboratories are working in this field. So you will find this TCGA, as I discussed, from this website, and what kind of data is available is listed here. You can have RNA sequence, you can have DNA sequencing, and you can have miRNA sequencing or this polymorphism data, and also now DNA methylation and RPPA data. So these are all data that are available from this data repository. And then you can have this CPTAC, clinical proteomics tumor analysis, where you can have proteomics data that are corresponds to this TCGA cohorts. So this is also interesting. So you can download the genomics data from here, and then proteomics data from here, and then perform analysis and perform many things to make some integration between these different layers of data. And then you can also have data from this International Cancer Genomics Consortium, and or there are also, I think, popular data sets like Target. Target is one of these pediatric cancers where you find gene expression, miRNA expression, copy number, and then sequencing data as well. And as I already talked about this omics discovery index, and where you can find data sets from different, even they are also directed to different repositories also. So you can have data from genomics, transcriptomics, proteomics, and metabolomics. These are all different types of data that you can get from this omics discovery index repository. And now coming to tools and methods that are available. So far we have just talked about from where we can get the data, but I think most of computer science people might be interested in knowing if there are what kind of different tools are available, or where are the opportunities where you can contribute to make sense of this kind of omics data. So far we have seen a really good growth in terms of availability of tools and methods. So basically this picture is showing you the tools and methods that have been that can be categorized on different approaches. For example, you can have there are methods that are based on networks. Then you have methods that are based on correlation, that you have methods that work on similarity indices, or you can have multivariate methods. You can have a new complete list of new methods that are basically focused on Bayesian approaches. And then you can have fusion approaches, and you can have, so basically their objectives are also different. So those in blue, they are focused mainly for disease subtyping, and those with these within the oranges, they are focused on disease insights, to get some insights into disease and understand the underlying mechanisms. And those in this greenish color, they are focused on doing the biomarker predictions. Right, so you can also find some cross overlaps between different categories. So for example, you can see that SNF is so for example, SNF is, you can find it here in this fusion category, and it also based on SNF similarity, sorry. And then you can have mainly this approaches that are focused on also network paradigm, or you can also have this main popular tools are basically this and net ICS, and then there is also omics somewhere, mix-omics. So this is also a very interesting tool. So I will show you some insights into this mix-omics, which is based on multivariate analysis. And my coming slides, I think I will talk about this. So you see there are a bunch of methods that are available. They're already there, and there are lots of data is available. So you can see that there is also possibility to, of course, to develop new methods and to improve these methods. But this with the availability of this kind of data, so a lot of work and opportunities has also become in terms of developing these methods, right? So most of these methods were existing from other works, but they have not been, or they were not been, you can say, directed or towards this kind of data. So a lot of people now have become interested, and because they are already methods that exist, they can also be used, for existing methods can also be used in this domain. So and also, as I said, there is of course no doubt that there are opportunities to develop new methods with which can help to handle or analyze this kind of data. So let me first talk about this strategy of handling omics data. So as I was saying, so you need to have a good strategy to handle this kind of big data. So you need to have good data management, and you need to have, for example, under this you can have good project metadata. So you need to basically define, or you need to have metadata, proper metadata, clear metadata to describe your data. And then you need to have metadata also on experiment. So how this experiment, how the biosamples were collected, how these treatments were used, how many reagents were used, what are different protocols and different subjects that are available, right? So then you need to have primary results data. So how this can be used, or what are the different results that are available with this data. So for example, you can have expression values that are available from this data. So you need to have a good strategy for also providing this kind of data. And then you also need to have this processed data. So you can have, for example, data matrix of fold changes basically, and p-values, fold changes in terms of gene expression also it is possible. And then you can have data processing metadata, what kind of normalization and summarization methods were used, and what other some other information about these approaches or processing that has been implemented. And then you need to have interpretation of the results, right? So you need to have which genes are interesting, which proteins are interested, and metaboloids, and so on. So we need to have some biological interpretation of the results. And then you can have this interpretation even on the metadata. So you can just say how we interpret the fold change, how do we interpret the p value cutoff, and what can be other measures that can be used, or they can be help to interpret our data. So this kind of things need to be clearly mentioned when we are developing a strategy for handling this kind of data. And of course, we need to have new visualization approaches to in context of biological pathways and networks. And before we go to mix omics, let me show you this what are different integration schemes that are available when I am saying multi-omics integration. So basically there are two kind of schemes that are possible. You can have a horizontal integration scheme where you have basically you have, let's say, this kind of matrix data is available in form of matrix. So this is just one example. So we have mRNA expression from N1 samples and P genes, and we have different studies that are available. So what is happening is now we have different set of individuals which have the similar kind of data, same type of data, but on the same genes. So this kind of data may be available, and what can be done is we can perform a horizontal integration. So basically this kind of data is available, or we are kind of doing analysis on the single level. Either it can be transcriptomics, or it can be genomics. So there are many meta-analysis approaches that are available that can be used for performing this horizontal integration of the data, and they can be then correlated or associated with this clinical outcome, right? So this is you can say one type of integration scheme. Or you can have vertical integration, right? So vertical integration primarily means that you are drawing lines across the different layers of multi-omics data, from transcriptomics to proteomics, from proteomics to metabolomics, and so on. So here you can have same samples. So you can say this same kind of one cohort, and you can have different layers of data from this cohort, or maybe from different cohorts. And what you can have is you can have P1 features from based on your, let's say, one layer of data, and P2 features from another layer of data, and P3 features from another layer of data, right? So now our idea is to kind of integrate this kind of data. So this can be performed in parallel. So this is called parallel integration. And then we can even have this hierarchical integration, where we integrate P1 features, P2 features, and then P1, P2 features with P3, and so on. So this is also one kind of scheme that has been followed up. And then, so yeah, this is what I was meant when I'm talking about this vertical integration, where you have so this is just a slide which compares this conventional reductionist approach with this transcriptomics approach, holistic view, we can say. So basically what we have is, in conventional approach, we look at single gene and then analyze and across different layers what is the mutation, what is the function, what protein is encoded by this gene, and then we probably look at where it is interacting with other metabolites. So we are looking only at a single gene or single molecule, single feature, or you can not feature but single molecule. So this kind of work has been very outdated, and people have been doing this before last 3 decades. And now you can say this single omics has also been in quite a while in last few years, where people were just focused on a single layer. So whether it is genomics, transcriptomics, or proteomics, they were mainly focused on only a single layer of the data. So the people were not drawing any conclusion from across these different layers of data. But now in this multi-omics or transomics discipline, people are looking across, not only within the layer, but across the layers. So this is basically the idea. So you need to have this holistic overview or holistic view of solving these problems nowadays, right? So this is what this slide is showing up. And then yeah, so this is just another example where people, you can say, just started with the same. So this is showing you different strategies of integration. So yeah, so one strategy is that you start with the same samples, for example, where you have grown your yeast organism in this flasks, and then you just do yeast lysate, and then you extract only the lipids. So your experiment or your aim is focused on getting this lipid profile, and you do the lipid extraction, and then you perform this mass spectrum analysis, and then you get the spectrum, or you get the profile or lipid profile in given sample. So you basically draw this graph and see which, to identify and quantify which lipid molecules or lipid species are expressed in this sample. On the other way, you can also, with the same kind of samples, you can perform, you can make a protein expression, protein profile, you can say protein quantification and quantitation. So you can you follow different set of protocol, and then you can also get similar kind of expression or identification and quantification for proteins. Now, so you can So now what is integration? So you need to now draw some conclusion why, given this lipid profile, why you are observing this kind of proteins expression, right? So this is basically will be your one kind of data integration. Now I was talking about this mix-omics, so I will quickly take you about this mix-omics method that I was mentioning. So basically it works on this, as I was talking about that there are several methods. One of them is based on this matrix factorization, mix-omics, where you have basically different layers of data and then you do this matrix factorization uh, on this input data, right? In case of Bayesian, basically what you do is you have some priors and then you infer the posteriors, or you can perform this network-based analysis, and you can also perform this single omics analysis, and then also you can draw correlation and overlaps. So there are different strategies, and the methods that I've shown you, they are based on these kind of approaches. Okay, so I will skip this. This just say what we expect from this kind of integration. So what I want to show you is this, those who are familiar with this R programming, they can have a look on this. So this is one package, R package that are provided, and this is called Mixomics. And this R toolkit is basically a multivariate data analysis and statistical integration of biological omics data. Basically, it specifically focus on feature selection and visualization. Under this tool also, you will find that there are different options. So, for example, it depends what kind of input you have. So, for example, if you have this singular data, single level data, so you can perform this PCA or IPC or PLS method. If you have, let's say, 2 layers of data, you can perform PLS or RCCA and so on. So if you have, let's say, multiple layers of data, so transcriptomics and proteomics, or you want to perform in this analysis in a horizontal fashion, you can perform it with the Diablo method that are provided in this toolkit. Or if you are interested in doing this vertical integration, that is so another approach. So you can perform this with the help of this mint option or mint tool that is available, right? So these are all multivariate methods. They are available in this toolkit, and also depending on what kind of data you have, you can also have different possibilities for the graphics to visualize your data. So for example, you can have this sample plots, or you can have this networks, you can have this clustering PCA plots, or you can have this three-dimensional overview of this your data, or you can also do a variable plots, or you can also create this circular circus plot kind of, where they also provide very interesting information. So these are all kinds of things that you can do with this Mixomics R package that is for omics. So what I will take you, let me check, I have still few minutes. So now how does the methodology, how does it works is very different from what kind of integration you are performing. So for example, as I was saying, if you have basically an integration means that your data sets measured on same n samples, but from different omics. So the same kind of samples, so same cohort, you have multimodality data on same samples, transcriptomics, proteomics, and genomics, and all they are available from same set of cohort, which is really rare from my experience. Getting such kind of data in practice is very difficult. At least at present. And then, sorry for this. So this Diablo basically works by this method. So you have n by p1, n by p2, and so on. N is constant. What is changing is the features, right? So and then you can just perform this matrix factorization to perform this analysis. And whereas in case of this MINT approach, where you look at so you have same features, right? So you have, but the individuals are different. So you can have independent studies, you can call it, where you need to perform this integration or from across the different studies cohorts. Those, so that is also possible. So you can perform, if you have such kind of data, you can work with this MINT approach. And usually, so the challenges here are that usually what we will see in biology is that our features are you can say very high dimension, whereas samples are really few. So usually we have hundreds, thousands of samples, and on most of the time millions of the features. So this is a big challenge. So we need to have this unwanted also systematic variations due to differences between platforms, lab, or protocols. So these are also very many challenges. I was also talking about these in the beginning when I was showing you this slide. Of course, there are many other challenges, but these are computational, and you can say this technical errors that are most frequently observed. And I was also talking about the single cell omics. So I think I don't have much time, but maybe some other time I will take you through the single cell omics data, where a lot of work is happening from this human cell atlas data, that platform data portal that I showed you. So many things can be done at a single cellular data. So you can have this trajectory inferences, you can have differential expression from one cell to another cell, you can do clustering, and you can also do compositional analysis to identify which genes are expressed in a given condition or in a given set of individuals, right? So these are all possible things you can do. You can study gene dynamics, right, in a given cell, and you can also look at this metastable states, where you see the cell is stable, or where you can see which states in the system or cellular system are stable, that are most frequently found in our biological system, right? So these are all many interesting things that can even be done at this single cell multi-omics data. So I think with this I will stop, and just before that I will give you some messages. So since we are entering this new era that first requires data-driven approaches to make sense of big biological data, and from a holistic view, we need to have this holistic view to a hypothesis generating approach, and we also does not include a prior biological knowledge in computational method. So it is important to include such kind of knowledge before we develop these computational methods. And then a new field are emerging that blends computationally savvy biological scientists and vice versa. And we need to have a pace with the techniques. So we need to develop new computational tools is important, because to understand or to advance our knowledge, such kind of computational tools are really needed. So with this, I thank you, and if there are any questions, please let me know. I will be very happy to answer those.